How does epigenetics impact brain development? Imagine your brain as a bustling city where thousands of roads connect different neighborhoods. Now, think of epigenetics as the traffic signals that control which roads are open or closed. This process influences how genes are turned on or off without changing the actual DNA. It's like a switchboard that helps guide brain growth, especially during infancy and early childhood. During early brain development, epigenetic mechanisms such as DNA methylation and histone modifications act like traffic controllers. They decide when specific genes should be active to help from neurons, build connections, and support the brain's ability to adapt. These controls make sure that the right genes are turned on at the right times for neuron creation, migration, and the development of synapses, which are the communication points between neurons. DNA methylation is one of the main epigenetic processes. It involves adding chemical groups to DNA, which can turn genes off or reduce their activity. This process responds to learning and environmental cues. When a child learns something new, DNA methylation patterns shift to allow genes involved in memory and learning to be expressed more. This is especially true in parts of the brain like the hippocampus and prefrontal cortex, which are key for memory and decision-making. Early experiences, whether nurturing or stressful, can change epigenetic patterns. For example, a caring environment can promote positive epigenetic changes that support healthy brain growth. On the other hand, stress or neglect can lead to lasting epigenetic modifications that might affect how the brain responds to stress or influences behavior later in life. Proteins like brain-derived neurotrophic factor, or BDNF, are vital for neuron survival and synaptic plasticity. Epigenetic changes can influence how much BDNF is produced, impacting brain structure and function during critical periods of development. If BDNF activity is reduced due to epigenetic marks, it can slow down brain growth or affect learning abilities. Enzymes such as DNA methyltransferases and TET proteins help add or remove epigenetic marks. These enzymes are especially active in neurons during early development and learning. They help keep a balance between activating and silencing genes, allowing the brain to adapt to new experiences while maintaining essential functions. Understanding how epigenetics influences brain development highlights the importance of early life factors like nutrition, caregiving, and stress. These factors can shape epigenetic patterns that last a lifetime, affecting cognitive and emotional health. This knowledge shows that early interventions can help promote healthy brain growth and reduce risks of future difficulties.